both received their PhDs from the University of Illinois. They both work in the same area of cognitive neuroscience and developing these optical imaging technologies for sensing the brain at the organ level. And I think they'll take us to the top of the brain.
Okay, so this talk is going to be very different from the previous one in that we are using optical methods, but in a completely different approach than microscopy or Raman spectroscopy. Here we are talking mostly about using images in these methods in a different approach, which we may call diffusive optical imaging. So clearly some of this figure was already presented before, so it's not particularly novel to you. This just shows that certain wavelengths are better than others to look at the brain. And first of all, perhaps I should first frame the problem. We are interested here in doing imaging of the activity or whatever of the human brain, and doing this in a non-invasive, completely non-invasive fashion. So we are not putting our instrument on top of the brain, we are putting our instruments on top of the head. And we are not making any lesion whatsoever, we are just observing the activity there without any particular lesion that we make. In addition, we cannot afford using dyes or any other instruments like this. So we are using intrinsic signals that come from the brain itself when the brain is active. So this is our problem. And for this, we go to a very different solution from the solutions we saw before. Of course, we need to sacrifice the spatial resolution that was presented before, and perhaps some of the temporal resolution, although not much, but arrive to a very different way of thinking about activity in the brain. So first of all, let's examine here the concept of wavelength to use. So all of you are here, and none of you can see each other's brain. And the reason why we cannot see each other's brain, of course, is because we have tissue on top of the brain, which is hiding the brain. This tissue is highly scattering. So the main property of this tissue is that it's very highly scattering. In addition, it is also absorbing. So the absorption, it comes mostly at the visible wavelength, comes mostly from hemoglobin. Although, of course, hemoglobin exists in different species. You can have oxyhemoglobin or deoxyhemoglobin, and they have slightly different absorption spectra. This is one of the way in which light is that hides our brain. The other thing is, of course, the scattering of tissue. Now, if we use near-infrared light, the scattering becomes the most important factor in preventing us from seeing the brain. So if we were just here using, instead of having the regular light of the various wide spectrum that we have here, we had only light that is near infrared around here or far red, we still will not be able to see the brain because of the scattering effect. So scattering is a problem, as it was for the multi-photon microscopy, but in fact actually it shows actually interesting properties for us. So how do we obtain optical images non-invasively? And the principle here is based on the basic principle of diffusive optical imaging, which is separating sources and detectors. So here, when we are present, the reason why we cannot see each other's brain, in part, is because we have a diffuse source of light that illuminates all the head. And this diffuse source of light makes the fact that the light will be reflected by the very first part of the head, and we will not be able to see beyond that, because everything else is trumped. However, if we, instead of using a wide source of light, we use a small source of light, just as is shown here in this example, and we just measure our light at some distance from the place where it comes, we generate a different situation. So the light that we are injecting in the head, and this will be a sort of a cross-section of the head of a human person. This actually comes from an MR image of a person. And you put the light here. The light will, because the medium is very highly scattering, the light will be diffusing all over the place. An example of what will happen for this is very similar to what happens on my finger. If I put the light on my finger, like here, you can see that light is scattered around. So this is a laser pointer. It should be very accurate and very nicely collimated. But in fact, when you put it on my finger, it illuminates the whole tip of my finger. This is because light is actually scattered by the tissue to such an extent that you lose completely the coherence that you get from the original image. You just get a very big blob of scattered light in all directions. At this thicknesses greater than a few millimeters, the medium can be, the process can be considered really a diffusion process. It's just photons behave like they are scattered to such an extent that the process can be well described using just diffusion equations. So this light diffuses in the middle, but of course this medium, if you had some photons that come here, there will be photons all over the place. Some photons that injected in a source here will arrive to detectors located at some distance here. Now it is interesting to note that the trajectory that photons follow between this source and this detector is not entirely undetermined. It can be described in a certain probabilistic manner. So the higher probability is that the photons that are emitted by the source and arrive to the detector will have moved within a certain particular volume, which contains the vast majority of these photons. There are very few photons that go between the source and the detector that travel very superficially, so very on this superficial line. The reason is because of the scattering, very random motion they have, sooner or later it's very likely that they will arrive to the surface of the head. And then there they will meet a medium which is non-scattering, which is air. So as the medium there is non-scattering, they will just start again propagating more or less in straight line outside from the head. So those photons never reach the detector. At the same time, photons that travel in other directions at the beginning in the random motion are also unlikely to get to the detector. So you are only getting a proportion of the photons that you presume injected at the source, and those photons are not really very completely equally representative of any possible location in the head. Rather, they represent certain particular locations. So they move through those particular locations. And this sort of migration of photons in this diffuse motion from the source to the detector down there determines a volume that is explored by any pair of sources and detectors that you can put on the head. Of course, the particular volume depends on where you put the source and where you put the detector. And if the sources and detector are located here, then the volume will be here. If you're putting them in other locations, like here, the volume will be down there. And also, in addition, the volume will go deeper and deeper depending on the distance between the source and the detector. So if the source and detectors are very close, you are going to see only a very superficial area, or mostly a superficial volume. But if the source and detectors are going very separated by a large distance, then this volume stretches out and it becomes sort of a larger arch. Okay, so this is really the area that is measured by anything by our system. So if we have a source here and the detector there, any measurement at the detector will be influenced by phenomena that happens within this volume, but not by phenomena that happens anywhere else. So this provides not as good a spatial specificity at all as the one we are getting from the two-photon microscopy analysis or from other type of coherent light systems, but still provides a certain type of of localization of the type of measurements that we are obtaining. And this type of localization can be further largely improved by not using just a pair of source and detectors, but using a lot of pairs that are superimposed in different ways, and then therefore can cross this any particular volume in many different ways. And through that, and using some type of three-dimensional reconstruction algorithm, we could generate some type of image of phenomena that happens within the brain. So this actually is a way of getting some type of image with a resolution of some millimeters that we can get from just measurements from the surface of the head. Now, of course, we need a lot of sources and detectors. So this is an example of one of our, two of our collaborators there, Cathy Lowe and Ed Macklin. And this is one of our helmets that we use to hold our fibers on the head. And so these are the various fibers. And the bigger fibers that you see are detector fibers that are bigger to collect more light. And smaller fibers, there are not that many that are very well visible, are source, like this one here. Those are source fibers that will carry the light through the brain. Now, for us, measurement of it is very critical, of course, once we are on the surface of the head, to know where these measures are coming from. Because as I said before, if we want to do a map of the brain, we need to determine where we are measuring with respect to the brain. So one process we do is a process called co-registration, by which we identify exactly the location of each source and each detector over the head of a particular person. In this case, we use an MR image to represent the anatomy of the head of a particular person. And this image, MR image, can be considered just as, in terms of the whole head, or can be considered in terms of only the brain that is inside the head. And of course, as I said before, we also do some type of image reconstruction algorithm, by which we can go from values that we observe from source detector pairs that are on the surface there. So this will be a source and this will be a detector, and so on, and there are many various combinations of them, and we can use these various combinations to sort of reconstruct the volumes that are investigated by each of them, and the crossing that we get for all of these different from different channels. A channel is a pair, a source detector pair for each particular for various locations, and we use this to generate an image, basically, of the brain of whatever happens to the light that we insert in the system and that arrives to the detector. Okay. Now, this image, of course, doesn't have the micron resolution that we had with multiphoton microscopy. Rather, we have a resolution of the order of millimeters, so 1,000 times smaller. We don't see individual neurons, but we see regions of the brains. And these regions of the brains, basically we are sort of summarizing activities for larger regions by fundamentally computing some type of large statistics across a large number of neurons or large number of structures. Now, as I said before, we have light coming in and light coming out. However, there are different types of instruments available that vary depending on what type of, what are the properties of the light that come in and the light that come out. Specifically, two types of instruments are being used in practice, in common practice, quite a bit. One is called CW instruments, which are by far the most common because they are the cheapest of the two types of instruments. And with the term CW, or continuous wave, really we mean that the intensity of the light is considered as practically as a constant. It's basically always the same amount of light. And what we can measure with this is just the variations in the amount of light traveling between a source and a detector, something which we call intensity. And we can measure these variations over time. we can use different wavelength of the light sources that we are using. The light sources are oftentimes laser diodes with some type of wavelength. And most commonly, the wavelengths used are in the near infrared or far red, like 690 nanometers, 850 nanometers, 830 nanometers, something like that. And many of these instruments are built, many of the instruments you find around using diffusive optical imaging are based on this particular type of logic. Another type of instrument is called a frequency domain instrument. These are much more rare, although that's the type of instrument we have in our lab. A frequency domain instrument is different because instead of having a light source which is constant in intensity or practically constant, has a light source which is modulated in the radio frequency domain level, hundreds of megahertz, typically 100, 110, 200 megahertz, something like that. Now, these frequency domain measure instruments are interesting for us because they measure not only the amount of light that goes between a source and a detector, but they can also measure the oscillation of light, the light that is emitted by a source, and how the oscillations is distorted before going to the detector. And it is distorted both in amplitude and in phase. Okay, so in amplitude, we call the amplitude oscillations, of course, we call AC intensity oscillations at the modulation frequency. And we can compute the ratio of AC over DC, and this will be the modulation ratio of the two lights. So how much less modulated is the light coming out of the head than the light you put in. The other part, the other measure is the important measure is a measure of phase. Phase is a delay of the oscillations that you do in this of the light that you put there. So this delay is of the order of some nanoseconds to picoseconds. And this is because the light takes about picoseconds to nanoseconds to move across the head and arrive from a source to the detector. Now, this is, of course, because for various reasons, one of which is because the refraction index, of course, of the brain tissue is higher than the refraction index of air, and is, of course, that for that reason the light goes slower. But much more, a much bigger factor is determined by the fact that the path that each individual photon take to move between the source and detector is much slower than is much longer than the path than the simple source detector distance. This is because of the various scattering phenomena and diffusion mechanisms that we were describing before, which basically slow down the movement of these photons through the head, just because they have to go around through a lot of very complicated paths. Now, the advantage of frequency domain instruments is that they allow you to measure the photon time of flight, or the time taken by photon to go between the source and the detector. This parameter is important in two different ways. In one way, it is important because if we want to use spectroscopic laws to determine the concentration of substances in a particular tissue, we need to consider not only how much the light is reduced, but also how thick a particular preparation is. So this is an important aspect of measured spectroscopic effects, because they will add up as a function of greater effects, absorption effects, because the absorption effect will add up as a function of distance. And therefore, we are going to need some measure of the length of the path that photons follow within the medium to really get a reasonable amount of measure of this type of an absolute measure of this of concentration of substances using this approach. Another reason why it is useful is because as we see later, these measures have very different, some very different parameters of across space, the phase measures, than the intensity measures, which are very useful for us to identify phenomena in the brain. So this is just a description of one of our instruments, a sort of a diagram illustrating how our instruments work. We have a very synthesizer that generates high frequencies, and these radio frequencies. And these radio frequencies are used to modulate the source of light that we are presenting. So the light of sources are modulated at hundreds of megahertz. And then there are our detectors. We typically, in our lab, you use PMTs. In our instruments, we use PMTs. Other instruments may use other type of detector, light detector systems. And there is a trick to be able to do all of this operation of measuring phase delay and see intensity very cheaply, and that's to add beating frequencies to the measurement, to the detector's instrument, so that we can measure everything at a much lower frequency, which is much cheaper for electronically. And we can use these to derive different measures. We'll talk about these measures more at length later on in this presentation. There are two types of measures that we typically are used in functional brain imaging. One is called near-infrared spectroscopy, and Monica will come back to this measure, which is really related to the concentration of oxygen deoxyhemoglobin and how it changes in particular part of tissues that are active. And another is related to directly to neuronal signals that can be detected. Also, they are related to changes in the scattering of tissue, and we find out that neuronal tissue, when it is active, scatters light differently, at least when we are using this in this sort of large statistical analysis level that we are using here at a macroscopic level of several millimeters level. So basically, we can use this method, diffuse optical method, to study many different things. The vast majority in the literature use investigate one particular thing, which is changes in the concentration of oxygen deoxyhemoglobin during a particular task, which is called near-infrared spectroscopy or an IRS. And basically is a measure of the changes in the level of oxygenation of the brain parenchyma. We, however, have developed another set of other measures that all can be obtained with diffusive optical methods. Some of these are measures of brain anatomy, related mostly to the opacity of different type of tissue, which are important per se, but can also are important for the three-dimensional reconstruction of the optical data. Then we have measures of physiology, which are basically not only a measure of the concentration of oxyhemoglobin, but measures of neuronal signals, something we have worked on for the last 20 years. Then we have also measures of the arterial pulse. So before there were talks about arteries and how you can measure with sonograms and so on, some parameters of the arteries. You can also measure them with these optical methods. And there are, in the end, also some measures that we have not used ourselves, but some other lab have been using, that use very high frequency changes in the activity in the signal to study the movement of blood cells, or red blood cells, through the cortex. So I will focus for my part only now on the measures of anatomical measures of opacity. The rest will be presented by Monica later on. The idea here is this measure we call opacity here, just what is an obstacle to the movement of light into the tissue, mostly due to scattering and absorption. Now, tissue opacity can vary a lot between different types of tissue. And this is related to the fact that different types of tissues have generally different absorption properties and different scattering properties. So, for instance, blood has a very high opacity because it is highly absorbing. It is like white matter, which is the part where there is the myelin, has a moderate type tissue opacity because the myelin itself is actually a substantially scattering substance. In fact, it's sort of white and pretty lucid if you look at it on a transection of the brain. It is, on the other hand, another type of brain tissue, which is the cerebrospinal fluid, or head tissue, cerebrospinal fluid, has a very low opacity. It is basically a clear type of structure of tissue and has very low absorption and very low scattering. We can measure tissue opacity by simply looking at how the light decays with this source detector distance. So this is actually a global measure taken across a number of channels and a number of subjects, and it shows that on this axis, source detector distance, and on this axis is a logarithm of the light intensity of the that arrive to the detector. And you can see here that there is clearly an inverse relationship between these two, and this being a logarithmic curve, this is an exponential decay, just as presented previously, and the light amount of decay decays with distance in a very characteristic fashion. Now, the exponent of this decay, basically the slope of this curve, is associated with the product of the absorption and the scattering coefficient. So if you compute the slope of this function from this you can derive the information about the product of the absorption and scattering coefficients. Okay, now this particular approach allows us to measure basically for various locations of the brain what is their tissue opacity. And the result, you can do a map of the tissue opacity obtained in this fashion. So this here to the right is a map of tissue opacity obtained, actually is an average of a bunch of subjects, 40 some subjects. And you notice that there are areas with higher opacity. Other areas, this map is thresholded, so other areas we don't show what is their opacity in these other areas. This is just a way of thresholding the image. And if you compare this image with a venogram obtained with MR of the superficial veins in the head, you'll find out that these two maps look pretty similar to each other. So in fact, actually, this opacity measure really is sensitive to the presence of veins under the particular measurements instrument. So this is because veins contain a lot of blood and contain much more blood, in fact, than arteries. So the blood stays in artery for only about a second or so, but stays in the vein for about a minute before coming back to the heart. The consequence of this is that there is much more blood in your veins at any moment in time than there is blood in your arteries, about 60 times as many. Okay, so because of this, the arteries are the main container of blood in your body at any particular moment, and they they absorb light of the wavelength that we are using quite a bit, which is about 690 to 830 nanometers. And the consequence of this is that you see the veins using this approach Okay, so you can do a map of where the brains are in the brain using this particular method. Now, we also, however, opacity changes not only as a function of the veins, but it also changes because of various properties of the brain tissue itself. For one thing, we know that it changes with age. So here shows that this picture shows how opacity drops with age in normal adults. But we also have changes during development. And in this figure here, we show the brain of a 21-week fetus. And you can see here that this brain shows a very large hole here. This large hole is what it becomes in the adult, the Sylvian fissure. So here this will be the frontal area, frontal lobe. This is the temporal lobe, occipital, and parietal here. There's not much of a frontal lobe and not much of a parietal lobe at 21 weeks of gestational age. But there is a big hole here, which is called the Sylvian, which becomes the Sylvian fissure in the adult. When the age progresses, and we get like a 30 weeks of gestational age at which some fetuses are born, and you get a premature infant that is born at 30 weeks of gestational age. And if you do an image an MR image of this particular infant, it's not very easy to do that. You see still the Sylvian fissure here as a relatively large hole here, much smaller than at 21 weeks, but still significant. If you then say look at 1 month of age after the normal regular term infant at 1 month of age, you will find out that this fissure now, this hole, has become sort of a virtual hole and is practically compressed all out. Now this means that there is a development of the cortex during these periods that takes over and fills these holes. That's basically a development of the brain, not only of the cortex but also the white matter. Now we can find in fact that as these holes is filled, there is less cerebrospinal fluid in the brain, and as there is less cerebrospinal fluid in the brain, the opacity goes up. And so if you look at premature infants and of various gestational age or various weight, you'll find out that as they grow, their opacity in the brain goes, in particular in this area, goes up, mostly because of the elimination of this structure, this hole here, where there is a lot of cerebrospinal fluid. In fact, actually, we find that this is a very good measure of brain maturation for these particular infants. And this finishes my part. I don't know if you have any question before going to one section.
Opacity actually decreases as age?
Yes, in adult, yes. In older adult, opacity decreases. This might reflect the fact that there may be a cell loss, and maybe there is some level of atrophy of the brain, and so there is more cerebrospinal fluid that is coming back in the tissue. The baby depends on which age. At about their regular 1 month old will be about here, and those that are prematures, very heavily premature, will be about here. Any other question? Okay, well, let's go to Monica.
These are not designed for women clothes. Yeah, which I don't have. All right. Can you all hear me? Okay, so let me move forward. And so as Gabriele was mentioning, one of the reasons so obviously we can, in humans, you can derive anatomy from, for example, structural MRI very well. However, one of the reasons to try and derive anatomy optically is because there are populations, such as, for example, preterm infants, who cannot go in a scanner very easily. And yet, you might want to study both function and other physiological characteristics within the incubators. And this gives us some means of understanding what this anatomy is like, because both arteries and veins follow fairly prescribed pathways. And so if you can image those and you can image the opacity, you can retrieve at least some anatomical cues that then allow you to link back function to structure, which is an important point, again, in some population where you cannot do anything else. And there are a number of medical applications for this. What I want to talk to you about now is some of the physiological aspects of the optical measures that you can do non-invasively in humans. Just to clarify, when you are looking at the brain, the human brains, there are a number of imaging techniques that are available and a number of signals that can come out. You can start with neural activity, and there are, of course, electroencephalography of various types, magnetoencephalography, and the event-related optical signal all measures neural activity. Of course, though, the brain is highly vascularized, as we have seen in the previous talk, and when the brain is active, there is also vasodilation in active brain areas because blood flow to those areas, and first there is an anaerobic response. The neurons respond without oxygen, but then they need oxygen, and so more oxygen flows to those areas. And then you have an increased blood flow because of that, because there is that's the hemodynamic response to neuronal activity. And in the end, what you have is an increase in oxyhemoglobin, because the new oxygenated blood that feeds the active area flushes out the deoxygenated blood from those areas. And this signal is much more than what the neurons consume, and therefore it's visible, and it's visible in a number of ways. It's measured with the blood oxygen level dependent fMRI signal, and it can also be measured spectroscopically with functional near-infrared spectroscopy. And I'll talk about this and the event-related optical signal. There are, however, other signals that you can extract optically because you can also image the pulsation of the brain, the pulsation in the arteries in the brain, which is in fact one of the largest optical signals that you can retrieve. And the reason to do this is, there are multiple reasons to do this, one of which is that in this progression there are pronounced age-related differences that impact our research, for example, on Alzheimer's disease and other diseases, where you're comparing younger adults with older adults whose vasodilation system is changed. And so imaging this is actually has fairly important medical applications, or at least research medical applications.
So in NIRS, which signal dominates more? Is it the oxy or the deoxy?
The oxy is a larger signal. But you see both, and I'll show you. The largest signal by far when you do optical imaging in humans is the pulsation, and in fact I will start with that measure. So why do we care about pulsation? Arteries pulsate, and arteries go from the heart to the brain, and we are interested in this case in looking at arterial effects in the brain, within the brain. Now, in young arteries, the shape of the pulse is prolonged, and the reason is that because their arteries are elastic, you have both a response that pushes the blood forward and a recoil response that pushes the blood backward. And these two waves at the systole and diastole respectively do not overlap. The effect of this is that you do not have a peak of blood pressure, which is a good thing actually, and that the time in which the pulse propagates is longer, which allows for more nutrient exchanges and other things of that sort, which is all good. When the arteries walls become unelastic, however, these two waves overlap way more, and you have basically a bolus of blood pressure, which can rupture the arteries. I mean, strokes happen because of that, hemorrhages anyway. And also, it decreases the time, the period of time in which you can exchange nutrients, and the neurons therefore suffer from that. The pulse can be seen very, very easily in an optical signal. This is a single trial. This is one pulse. It's a large signal. This is AC intensity, percent change. This is the peak systole and peak diastole. The reason, so at the diastole, when the blood vessel is smaller, you basically have there is less absorption of light, so there is more intensity. And at the peak of systole, there is more blood flow, and therefore a more oxygenated blood flow, so the light is absorbed more, and you can see this. And you can clearly see the shape of this, which, as we know, and this has been used in Doppler, with Doppler sonography and so on, the shape of the pulse can be examined for diagnostic reasons. You can see here, if you do this is now real data. If you look at the peak of the, starting with the peak of the diastole, you can see the large arteries appearing in this diagram as they pulsate. This is time in milliseconds. So this is exactly this graph here. And you can see the large blood vessel appearing on this graph because they are pulsating, and so more and more oxygenated blood gets into them. Now, this is data again from, yes, those is all optical, done with diffusive optical imaging at the head. And in fact, we have the two largest instruments in the world here at Beckman. One is in our lab, and the other is a shared instrumentation funded by NIH that sits in the biomedical imaging center downstairs, and both have full head capabilities. This is the type of montages that you can use, and therefore you can really image the whole entire surface of the cortex. And all of these can be easily synchronized with the R wave of the EKG, so that you know you're talking about the same pulse. All of this is done here. Nope. No, what we do, it's I mean, there are people who are easier to image than others, but what you do, the sources are really 400 microns, so they go in between hair. The detectors are about 3 millimeters, and what you do is, through the holes of the helmet, you comb the hair out and you lower the detector fiber, and then you tighten it so that it stays with the hair surrounding it, and that's how it's done. In babies and preemies, you don't need to do any of this. You can put the hair. The hair is fine and usually almost non-existent. So these are now images. This is an MR arteriogram, so we are talking about arteries now. And this is an arteriogram averaged across a number of subjects done optically. And as you can see, there is a clear correspondence. This is an average. It is a single subject. This is done by looking at where the pulsation is greater, because the bigger the arteries, the bigger the pulsation. So you can threshold these maps and highlight the big vessels, or non-threshold them and get a complete map of pulse. And I'll show you some of those later on. The other thing you can do, and all of this can be done concurrently, the other thing you can do, and this is now a latency map, you can look at pulse wave velocity, which is actually a very important index of arterial health. It's used, again, as I show you before, the longer the pulse, the better it is in terms of health. And so the red is a longer pulse wave velocity, and this is a shorter pulse wave velocity. And you can examine this. And in fact, these colors here are corresponding to the places of exit or entrance of, for example, the middle cerebral artery. This is the emerging place of the middle cerebral arteries in this hemisphere. This is another way of doing the same map. Basically, you can see that you can extract these values, and we know that these values possess some meaning because, for example, we know that people who have a shorter duration of the pulse in the middle cerebral arteries are also those who perform more poorly in certain specific behavioral tests. For example, verbal fluency, Broca's area, the center that governs our fluidity of speech, is here. And because if you have a shorter a shorter transit time. Basically, if your arteries are stiffer here, then you will be having more trouble performing this test. So there are clinical correspondence to this with data. The other thing you can do is look at complete maps. And these maps, this is one version of this map. You could also map out the large arteries. But this kind of summarizes all of the arteries within the arteries and arterioles within the brain. This is the brain seen from the top. The nose is here. The ears are here. What we did here, these are people that vary in two parameters, age and cerebrovascular cardiorespiratory fitness. Cardiorespiratory fitness is known to improve all sorts of cardiac and also brain function. There are a lot of studies showing that people who are more highly fit basically have more preserved brain tissue, have more flexible arteries, and so on and so forth. So what you have here is someone who's young, and 10 is a very large number. So someone who's younger, relatively speaking, these are all older adults, but highly fit. And this is someone who's older and fairly low fit. And you can see that red here is less elastic and is bad. You can clearly see at a glance that there is quite a bit of difference in arterial stiffness across these four individuals. So you can see this data at the individual level. But the other thing that is interesting and we are exploring is the fact that the stiffness is not the same everywhere. And so you could actually, in terms of sort of the future plans and grand scheme of things, you could try and intervene on specific areas that have specific problems. Like here is a very high level of stiffness, the right parietal lobe. It's an area that might lead to strokes, and might be prone to strokes, lead to hemi neglect, and so on. So you could start thinking of intervention strategy if this becomes a diagnostic tool, because you would have the regional specificity to do that. And means like, there are arterial imaging tools, of course, out there. They are invasive, most of them. This can be used for research, which is important. The other thing is that, like Doppler, for example, intracranial Doppler is not easy. It will show you some large vessels and not others. This kind of gives you an overall measures of stiffness that then could be explored more with other tools. And in fact, in many cases, it could be done concurrently with other tools, because this is optical, so it doesn't have any counterindication for doing this. Just to give you an idea of the, yes, go ahead.
How do you separate out the compliance from the arteries from just that of the mechanics of the brain tissue?
So for now, it's a summary measure. We are trying, but you have different parameters. So you have the peak, which is basically the pressure. You have the shape, which is, I think, a measure of sort of compliance, if you want. And then you have the velocity, the transit time, which, again, it's a measure of elasticity. So you can start doing those kinds of things to separate the parameters of the different parameters. And you can also try to compartmentalize these measures. This is not done that way. But you could start thinking that if we have an MR arteriogram, superficial arteriogram, we mask the deep vein, the deep arteries, because we are not going to see them. Then you can mask those out and start seeing the arterioles in the smaller places and so on. So you can actually start doing compartments. And there are things that are too superficial to be, again, the brain. So there are a number of ways in which you can do this. Just to give you an idea of the types of data that we are getting, and this is really new stuff. What you have here is cardiorespiratory fitness, and this is compliance in this case. And what you see is that there is a healthy correlation between the two. And compliance, so this is still compliance here, predicts brain tissue preservation. So people who have more compliant arteries tend to have more total cortex volume and more subcortical volume. So it doesn't only influence the cortex, it influences the health of deep cortical tissue, including the hippocampus here. And then, especially with the white matter, the relationship is quite strong, because the white matter is not as vascularized as the cortex. And so it depends on these big arteries on the cortex to be. These numbers.
So how you get the total white matter from?
Oh, these are, so we use publicly available software called FreeSurfer that extracts, so we have an MR image, a volumetric MR image for each person. And then you can measure a segment in brain and measure all of these factors. And in fact, what we are heading towards is looking at specific regional estimate of volumes and specific feeding areas of these arteries done optically and seeing the regional effects of these. And also we have at the same time functional effects. So one of the things I haven't said yet is that this comes for free. So pulsation, if you do the experiment right, is there, and if you want to do brain imaging, which I'll show you next, it's an artifact because it's a big signal that it's not a signal from the brain, it's a signal from the arteries. But you get it anyway. So basically you can get concurrently some anatomical measures done with opacity. So you get your veins, you get your arteries, and then you get some imaging functional measures, the spectroscopical measures, and then IRS and the neuronal measures, so that you can get them all concurrently, which has the advantage that you can look at, for example, neurovascular coupling or other relationship between them, which are otherwise are very difficult to do because they're not necessarily within the same volume. Yes?
Question, so this arterial compliance, what part of the brain are located?
This is overall.
Overall?
Yeah, but we have it regionally, just lots and lots of data.
And
Yes.
then if you look at the graph or some of the big ones show, there seems to be, I mean, not much of the statistics, but there's some people who have, like CRF of 10, but they have like less compliance with people who are.
Yeah.
So what do you think explains those relations?
There are, so in here, age will be a part of this, right? So this is not with age, this is with CRF. CRF can be higher for people that are older, but older people might have still less compliance because they're, so there are multiple, all of these, when you go from tissue to the system like the previous talk to humans, there is a big leap because in humans you have a tremendous multifactorial system where you have multiple contributing causes. So you can eliminate some, we eliminated gender here because the size of lungs of women is smaller. And so if you look at raw numbers, that needs to be corrected because it's obviously women don't live less than men, but they have smaller lungs. So you have to sort of titrate these values. So there are multiple contributors. Diet is another one. There are genetic factors that can contribute to this. And there is age. And again, if you, I don't have the slides here, but if you look at the relationship between this and age, there is obviously another correlations. And what you can do statistically, and we have done that, is to do mediation analysis where you put multiple factors there and see how they basically contribute, or multiple regression types of approaches or other things to see the relative contribution of this. But the advantage, just like we were talking about before, for example, if you clear the brain, of course you can see through much more, but then you clear part of the activity. In humans, if you take out all of these factors, then you do an animal experiment where you control them much better, but then it's not a human. So, I mean, these are the pros and cons of doing research at different sort of levels that I think you have to live with. And this is obviously, these are not going to be straight line. This is not going to be a perfect fit because there are always multiple factors fitting there. Nonetheless, these are sizable correlations for these types of data. Any other questions before I go on? Okay, so now premature babies, and this is where we are going with this, tend to die or anyway have various serious disabilities, including cerebral palsy, because they tend to develop hemorrhages because their arteries are not stiff, but their arteries have not developed elastin yet, and they're outside the womb, and so they're subject to this. So, and there aren't too many ways, again, of imaging these babies because they stay in an incubator. So what you see here is that we can image actually these babies with optical imaging. And you can also see the latency of these pulses. And the hope here, and these are data right off, out of the press. We are still analyzing them. But the idea here is that you might be able to predict some of these hemorrhages. And in babies, because their brains are so small and not gyrificated and not much myelin, you can really see it almost through the ventricles. And most of the hemorrhages are within the ventricles. So we are analyzing data now where we have a cohort of preemies that have, some of whom have had hemorrhages. In fact, so this shows you that this is a fairly reliable signal, even though, again, the spatial resolution is not fantastic. But what is more important here is in this. So this is a score of severity of intraventricular hemorrhage. And this is the number, the number of premature infants, and this is the elasticity measured with the pulse transit time, basically with optical. And you can see that the babies that have higher elasticity, according to our scores, have very low risk or have had no hemorrhages, while the babies that have lower elasticity tend to have more hemorrhages. So you see these things, and of course this is a correlation, but the idea is that maybe you can develop monitoring tools or predictive tools so that you can prevent some of this from happening. We are developing, so just to show you some, I have a slide coming up that will show you a picture, but we are also in collaboration with John Rogers here, we are developing some wearable electronics that will make it more possible to monitor these babies long term, so that instead of having to mount fibers on their head and so on, which is still cumbersome, you might have some monitoring, better monitoring tools. Any questions on this? So I'm done with the sort of artery, and now moving on to the measurement of oxy and deoxyhemoglobin, which is functional near-infrared spectroscopy. So just to give you an idea, again, in terms of scale, all of the techniques, this is sort of neuroimaging techniques classified by time and by temporal and spatial resolution, basically. Anything below this line is invasive, so it can be done in tissue, animals, or whatever, but certainly not in humans. Everything above this line, of course, will possess a lot less spatial resolution because it's in humans and it's non-invasive. Near-infrared spectroscopy is about here, so it has the temporal resolution of more or less fMRI because it's based on the hemodynamic signal that rises at that speed, and the mapping goes. Basically, it's a regional mapping. It's not a column or anything of that sort. Okay, so near-infrared spectroscopy is based on the fact that if you do a multi-wavelength spectroscopic approach in humans, you can locate your wavelengths at each side of the isosbestic point for oxy and deoxyhemoglobin, and therefore, with appropriate equation, you can image, have a spectroscopical measures either of the absolute or more typically relative amounts of these substances in response to stimulation. So in this case, the time is seconds, and this is oxyhemoglobin concentration in arbitrary units in this case. And this first part is baseline, and here this red rectangle is when visual stimulation started. So there are 20 seconds where the subjects looked at a reversing checkerboard. So it's a very powerful visual stimulation. You can see, just like expected, you see oxyhemoglobin going up and deoxyhemoglobin going down. And that's a typical hemodynamic effect. Where fMRI basically convolves these two, and it's more heavily weighted toward the oxyhemoglobin. In this case, you can look at those separately. So, for example, if you then again look at all individuals that are either young, highly fit, but older, and not so fit and old, you can see very big differences in these parameters. So you can see that young adults and older adults who are highly fit have a nice oxyhemoglobin response to stimulation, while the older adults that are not highly fit don't respond much, at least in terms of additional blood flow, just because their dynamic range is probably already dilated at the maximum and nothing more can go there. And this is the oxyhemoglobin, and these three are not significantly different, but are scaled in the right direction. So, just to summarize about near-infrared spectroscopy, which is pretty much the most common of the functional neuroimaging techniques used in optical techniques used, you have sort of a good to medium spatial resolution. We're talking about humans, so it's in the centimeter, high millimeters to centimeter range. You get separate measurements of oxyhemoglobin and deoxyhemoglobin, and you can potentially, depending on how you do the experiment, you can get absolute measurements, which is useful in some cases. Can be easily integrated with other technology, and I'll show some example later. You have fewer experimental limitations than fMRI because you're not laying in a scanner, you're sitting in a chair, and it's much easier, therefore, to accommodate in terms of positioning and other things. And therefore, because of all of these reasons, it's more accessible to special populations, including infants and children, older adults, and so on. Of course, the penetration is limited, and this is something important to consider, that it's a few centimeters from the head, from the top of the head. So you're talking about 2-3 centimeters, which will encompass the cortex, but certainly doesn't get to the middle of the head, where, for example, the hippocampus or the basal ganglia might lie in an adult. In a child, that's different. And the temporal resolution, because of the nature of the signal, this is a hemodynamic signal, is the same as fMRI. Now this is the last piece of my talk. We are going, how much time do I have? About 5 minutes? Yeah. We have neuronal signals here, and so the event-related optical signal, the fast optical signal, which is the same as this, except that instead of being averaged to an event, is analyzed in the frequency domain, and then some analysis about connectivity between areas. So the event-related optical signal allows you to image neuronal activity, and it's based on the idea that light scattering will change with neuronal activation. Let me skip this. Well, this is just to say that because it's neuronal, the timing is now in milliseconds, and the resolution is about the same as an IRS, the spatial resolution. So let me see if I can get this video to go. So when you look at a neuron being activated, you will see, let me do it again, that the shape changes, and we have seen some of that in the previous talk. See that the neuron swells a little bit. And the reason it does, is because as ions flow into the cells being activated, water does too. And so there are changes in terms of shape that occur during activation. And you can see this here. This is an image at rest, and this is an image that has contrast. Excuse me. I might need. Can you get me a little bit of water, Gabriele? You can get some contrast enhancements here. Sorry, I need to drink. I'm completely out of voice. You get the yellow signal here. No, I need to wait to drink a sip of water. Any questions in the meantime? Oh, thank you. Thanks. There is some. This is these data indicate the displacements, and also this data. If you see the dot here, I know it's hard to see from the back of the room, but you see you see displacements. I don't know what happened to my voice. I'm having an allergic attack of some sort, I think, because I have water, but so just to finish this. So it's also known that, again, you can visualize both compound action potential and, in fact, activity in the hippocampus optically in animals. This is with implanted fibers. This is with the fiber near the neurons being activated. What we think happens in humans is that the photon paths are changed when a large population of neuron is excited, so that at rest you have more scattering and less delay, and in fact more intensity. And when the neuron is active, the pathways become slightly longer. So you have a phase delay, basically, if you do it with FD type instrumentation, which is what we have. And so you see these tiny effects, which are in the picoseconds range, but yet are measurable, that signal that the fact that the area has been activated. And these are measurements, for example, in children. This is, again, a child. This is a premature, preterm infants inside the incubator. Now, the stuff that is on the face is not ours. It's just because they have to be monitored in a number of ways. This is the fibers that are not there for always. They are there for a little time to do the monitoring. And just to give you an idea of the data that we can get, this is one individual baby, a premature infant, and you see time here is in milliseconds, so this is 0. And the baby is being presented with a little speaker inside the crib. They're being presented with syllables, speech syllables. And you have seen there is a response in auditory cortex, which was the previous response, and now in frontal cortex. And we know that even at 28 weeks or 30 weeks gestational age, this baby can distinguish between sounds that they're expecting and those that they're not expecting, or the mother's voice and the nurse's voice, or those kinds of distinctions. So this is a fairly powerful system to image babies inside, again, an incubator when they cannot really be imaged with anything else. These are in again preterm infants. You can again see the deviant versus standards effect, and you can also responses at all trials in various cognitive paradigms. And so this has two sides. On the one hand, with this, you also get the pulsation, and so it might be a clinically relevant measure for this baby to learn how to prevent hemorrhages. But you also are studying cortical development, which is really, really hard to do in other ways. And you do it in one pass, non-invasively, within the incubator, so you don't expose these babies to any additional sort of stress or risk. There are, as I said, there are three types of signals that we can look at. There is the near-infrared signal, there is EROS, and this one is oscillatory activity. So you can look at rhythms because, as you probably know, in electrophysiology, you can look, for example, at alpha rhythm and so on. You can do so optically, so this is a spectral analysis in the alpha range of optical data, and then you can localize where these signals come from, and this is a 3D reconstruction of the data, so you can get an idea of what an adult kind of setup looks like. And as a final sort of slide, optical imaging is interesting because you can easily combine it with other methods. We record EEG routinely together with the optical data because it gives us an anchor for something that is like a gold standard for temporal resolution. You can do it in the scanner. We have acquired about 60 adults in the scanner with a forehead probe. So it wasn't full head, but it was a forehead probe within the scanner because the fibers are glass, so they don't interfere with the magnetic resonance imaging recording. And you can, as long as you have 15 yards worth of fiber, which of course are very expensive, then you can measure from outside the room and just carry the data outside the scanner. Similarly, you can do it with transcranial magnetic stimulation, which is a technique that allows you to create what we would call a temporary lesion in a human brain. So it either, depending on how you do the experiment, you can either excite the brain or inhibit the brain from outside. This is not easy to do because basically the TMS coil, in order to function, has to be very close to the surface of the head. But we built a system where the fibers are flat so that we could actually record underneath the coil, which again, it's an important thing because not many other systems can do this. And these are the data. And so, for example, this is a TMS pulse to the left motor cortex, which means that you see a twitch in the right hand or in the right index finger. TMS is transcranial magnetic stimulation. It basically there is a coil shaped like this that creates a magnetic pulse that is given to the head of a person. And this is single pulse TMS. And what it does is it creates activity underneath the coil that you can control. And depending on how you do the experiment, you can do multiple pulses, which will inhibit the area for a certain amount of time.
Response to the magnetic field?
Yes, the brain responds. And in fact, you can see here, this is a time again in milliseconds at 0. This is when the pulse is given. We have a point at 8 milliseconds, so immediately the recording time point after you see the effects underneath the coil. And what you see, though, is that the effects underneath the coil spread to the opposite hemisphere after a while, which is a way of studying, again, connectivity, because you then figure out which area is connected to what, depending on where because you have control of where you start the stimulation, but you can see where it ends. And so this is the type of experiment that, again, enables a number of new things, because there is nothing else that you can put underneath the coil that will not be affected. If you do EEG, it will have a huge artifact, because this is electromagnetic energy that you're putting in. And so this is a way of sort of imaging concurrently two things at once.
Question, is that the ERE? The event-related desynchronization?
No, this is event-related optical signal. So it's an optical signal underneath, and there is a twitch, so it's an activation basically, and it's entirely optical. These data are all optical data.
I'm still confused. Why the brain would respond to a magnetic field?
Well, if you think about, I mean, it's electromagnetic, so it's a zap. Yeah, it conduces current. Yeah.
Because I thought that people who study these things don't affect the brain. Maybe it does.
Well, this is a much stronger pulse than a cell phone. It's a Tesla. Yeah, it's, in fact, you have to find the appropriate place to put something like this in Beckman. It's not, it cannot be near outside walls, because it could interfere with someone with a pacemaker that comes by. And so you have all sorts of limitations. So just to summarize, the neuronal signal have good spatial resolution, reasonable, probably about 5 millimeters, I would say, the typical. They give us direct measurement of neuronal activity in humans, so this is postsynaptic activity for the most part. Can be integrated with other technologies, and have not that many limitations in terms of experimental paradigms. So again, a person can sit in a chair. The main limitations are the limited penetration, the low signal to noise ratio of it requires many trials. And of course, it's still under development, but there are now more and more labs that use this. And of course, there are, as I kind of mentioned during my talk, a number of different applications. So just to summarize for the talk, we hopefully have shown you that non-invasive diffuse optical imaging allows for a number of different physiological measures to be recorded, anatomical, hemodynamic, and neuronal. Most can be taken simultaneously, if not all. It depends on how you do the recording, but pretty much you can do them concurrently. And can be concurrently recorded with EEG, fMRI, and TMS, which are the standard sort of tools for human research, and in fact, in some cases, clinical practice. Can be applied to a large number of clinical population. We don't know of any limitations. In fact, we have some research going with cochlear implanted patients who cannot go in the scanner, but of course the fibers, again, don't interfere with the cochlear implant, so we can image recovery after the implantation with these methods. Of course, the main limitations are the penetration, because that will not go beyond the cortex, the outer layer of the cortex, except in premature infants. The limited spatial resolution, because compared to the methods that we were looking at in the two previous talks, this is limited, but it's limited to what it's limited in humans. And then there is low signal to noise for the neuronal measures, so you have to average a number of trials. And I think this is it. This is collaborators and the phases to go with them. Thank you. And my allergy is completely gone. I don't know what I breathed, but I went completely. Questions?
So has this been used successfully, or like even just whatever experimentally in like disease models for like epilepsy, Parkinson's, Alzheimer's?
No, we haven't done that yet. This is being, we have a fairly large enterprise going in aging, because in some respects you have to establish what the baseline is for aging. And we are now looking again at these arterial effects, and even in normal aging there is enough data now, in general, not just from our lab, with other methodologies suggesting that these arterial effects develop over a number of years. In fact, even in young adults, you can see some depending on the level of fitness and genetics and so on. And so the idea is that maybe this could be eventually clinical tool to sort of predict what's your trajectory and make correction early enough where you don't see Alzheimer's. You can correct certain things before they happen. One of the things that is interesting is that there are more and more data suggesting that in Alzheimer's disease, the vascular is crucial, so that even the same numbers of plaques and tangles, if you have a very good situation with your vascular, you're not going to show the effects of Alzheimer's, so you show them a lot less than if you don't. And so predicting, again, the health of the vascular thing is going to be used.
That's what I was thinking, also maybe the amyloid would affect the elasticity also.
Yes.
It would be already extreme.
Right, that's
Yes.
the sensitivity of the
Yeah.
So no, the scientists we have.
These are all healthy adults, so they have had nothing actually. They have had no strokes, no nothing. You want to start with a healthy sample, see if you have sensitivity to see even anatomical changes in a healthy sample. Of course, if you take an extreme sample, you're going to see much larger effects, but this is the baseline basically. And if it were to be a screening instrument of some sort, we would want that. I think you want to know what the normal is. Yes.
Interesting. You mentioned newborn boys potential. How big is the potential? How fast?
And the speed depends on the flow of the, so if an action potential is the duration of an action potential, if the ions, the water flowing in. How big it is, the slide that shows, let me see if I can get the, this is an idea of the scale. So this is the edge effect. The yellow thing is what I was able to talk, but this is the edge effect. The color part is where the expansion occurred. And this dot and this little other dot to show you the movement. But remember that what we are looking at in humans is the 3D trees that are, much larger than just the axon, and of populations of neurons that would create a displacement of some sort that might show these effects. The other thing is that there may be other things that some, to the extent that the glia is the same timeline, that might show effects. To the extent that membrane and refractory properties change in the same timeline, they might add up. So we don't claim that this is the only effects, but Rector has done studies in animal models showing that the biggest contribution is from sort of swelling of the cells. Yes?
So in general, I see there's a lot of spread in your data. The R-squared values are relatively low in the cross-section. I wonder if you see the same thing longitudinally if you follow an individual?
Yes, we have. We had a 2-year grant, so I'm trying now to get a 5-year grant for the same thing. This was, and we looked at the same individuals, year 1 and year 2. The interesting part is that the sort of stiffness in year 1 predicts how much the drop is in neuropsychological tests in year 2, in some neuropsychological tests. So many of them don't change in a year because these are human, healthy humans, so they're not going to, show, and it's not a huge sample, it's about 50, 60 people, because that's the type of, this work is quite expensive in terms of, not probably a Ferrari for the lens, but but close. I mean, this stuff still costs quite a bit of money. But so there are predictive effects from one year to the next, and of course the trick is to look ideally over a period that is longer, like 5 years in older adults would be a good range, because some of them unfortunately might develop some things, and so you can see them over the course of 5 years, see how that goes.
But here to consider that in all of those anatomical changes, the biggest factor is going to be genetics.
Yes.
And individuals come in with different brains, different sizes. In terms of their
So that variability is very, very normal. So even if you take a younger adult or anyone, and the variability is not random, because we have done studies where we link behavioral responses in tasks with anatomical effects and functional effects. So you can kind of go full circle and see that, for example, we were talking about the corpus callosum. If you look at the size of the corpus callosum, that's determined by myelinated fibers, which are largely cross inhibitory between the two hemispheres. And you see that the people, regardless of age, so this is entirely explained by the size of the corpus callosum, will have different functional effects depending on if they have small or large callosi, and also that they will have more or less cost in terms of slowing of reaction times with the same thing. This is data we published in 2009, I think. So you can see these things, but again, the level of noise and the imprecision, the factors that you're not accounting for in humans is way different than what you do even in mouse, where at least the animals are all genetically, probably manipulated, and at least genetically identical to one another. This is not the case here. Right? Any other questions?
Have you tried symptoms after the surgery for the nose? For example, you reach the brain,
we talked about this. I don't know, for the level of work that we've done, I'm not sure it's worth it, but it might become if, for example, people wanted to image the velocity of arteries that are more in the middle of the brain. Enrico Gratton, who's Gabriele's brother, is a biophysicist. We were talking about combining, in fact, photoacoustic effects through the nose, or because you don't have to go to the scalp, you could throw in the mouth or in the nose. But for research purposes, now these are volunteers, right? So these are volunteers that come in for non-clinical reasons, and so I don't know that I would want something stuck in my nose. But if if this became clinically motivated, then the states change and the things that you can do or not do change if there is a clinical benefit to the participants. But we haven't done it yet.
Let's thank those speakers again.